Right on time at 2 o'clock. All right. Well, thank you so much for everyone who has joined us so far and has agreed to spend their Thursday afternoon discussing the safety and science behind COVID-19 vaccines. My name is Laura Dirks, and I am a public health specialist here at SDSU. I will serve as the moderator for today's discussion. We have an amazing panel assembled who will be talking about the various aspects of the COVID-19 vaccine, why vaccination is important to ending the pandemic, the vaccine technology and development, and also the science and safety of the COVID-19 vaccines. Today, we'll have each of our panelists give a brief discussion or presentation about their topic, and then we will move on to the question and answer section at the end. So please, as you go through, if you've got any questions, put them into the chat box, and we will direct them to the correct expert at the end. The goal today is just to facilitate a discussion about the science of the COVID-19 vaccines and answer any questions about the technology. All right. Before we get started, are there any questions about from our panelists about the discussion?
Nope.
Awesome, great. So we'll start with Dr. Aaron Hunt. Dr. Hunt has completed both his MPH and his PhD at the University of Nevada in Las Vegas. He specialized in environmental and global health, and his background includes HIV prevention, global health, nutrition, health education, and environmental health. Currently, he is the MPH program coordinator and also an assistant professor here at SDSU, where his current research focuses on improving maternal and child health outcomes and reducing health disparities. So please welcome Dr. Aaron Hunt.
Thank you, Laura. I'm just going to share my screen, and I'll go through a short talk on herd immunity and why vaccines are important. Second. Can everybody see this? I'm hoping. So why vaccines are important to ending the COVID-19 pandemic, and we're going to cover herd immunity. So again, my name is Dr. Aaron Hunt. I'm one of the assistant professors in the MPH program in the College of Pharmacy in the Department of Allied and Population Health. And like Laura said, my background is in global and environmental health, kind of with a focus on HIV prevention. Just make sure everyone can hear. Perfect. So, and as a public health professional, this has been one of the biggest public health issues in recent memory. And I'm sure we've all heard a lot about epidemiology and public health and prevention. So I think it's an important topic. I'm glad you're spending the time today. And as these vaccines roll out, it's going to be pretty important that we have a high uptake. So hopefully this information will be useful to you. I just wanted to go over some of the recent rates and trends, COVID-19 in the US and in South Dakota. So as of yesterday in the US, there was over 26 million cases, confirmed cases. And over the last 7 days, there was about 1 million new cases. So you can see we're still having a lot of people being infected. Again, we're over 400,000 deaths in the US since this began in March. On a positive note, we have started vaccinating people. We're up to 33.9 million doses administered. So we're moving in kind of the right direction with vaccines, and hopefully our cases and deaths will start to decrease as those numbers continue to rise. And everyone's heard flattening the curve. We talked about this for months and months, why it's important to social distance, wear your mask, wash your hands. So you can see the big spike, around December, January. That's where it was kind of the highest. And now our cases for the overall US, they're going the right direction. So we're slowly decreasing. Specifically in South Dakota, again, we've had over 100,000 cases in the state. Right now, there's over 2,500 active cases with over 6,000 hospitalized, almost 2,000 deaths. And in the state, we've had about 76,000 people vaccinated. And of those, 33 have have had the 2 doses, which is required with the current vaccines. You have to get both doses. And this slide is from Bonnie Spector, one of the faculty at SDSU, and she's been sending this out. She's just analyzing the DOH statistics. So you can see our rates have been decreasing too. So you see the orange line at the bottom of this. This is the number of cases per day since March till, up till February 4th or 3rd. And we're decreasing too. And if we can get below that orange line, our substantial spread. So right now we have a lot of spread in the community. Once it gets below that orange line, it becomes less likely to get infected from spread within the community. So that's kind of the goal, to get underneath that line and stay underneath that line. And hopefully things keep decreasing. That's the goal. When we look at Brookings specific, for our county, we've had almost 3,500 cases. Right now, there's 128 active cases, 120 people hospitalized, 34 deaths, and about 5% of our residents have had at least one vaccine, and 2.5 have been fully vaccinated. And again, this is kind of that same slide. We want to be under that orange line towards the end to get underneath that substantial spread. So while we're moving the right direction, we still have a little ways to go to reach that. This chart is just showing the number of people vaccinated in Brookings versus South Dakota. So again, fully vaccinated Brookings, we had 2.5, and South Dakota about 3.5%. And this is going to be important when we get to the herd immunity, because we want to get to about 70 to 85% of people that are immune. So why is it important, the vaccines? We still need to wear face covers, social distance, and get vaccinated. Our cases are reducing. They're moving in the right direction towards that, getting underneath substantial spread in the state and the county. But right now, the majority of people are still at risk of becoming infected because they either haven't been infected or they haven't got the vaccine. So I'm going to play just a short video about herd immunity. If you're familiar with herd immunity, this will be pretty basic, nothing new, but if you haven't heard of herd immunity, it's a pretty good video to just do a quick overview.
What is herd immunity? Picture a community or herd of healthy people. An infectious disease would pass easily through the community from person to person. Vaccines train our immune system to protect us from diseases it hasn't come into contact with before. For example, the MMR vaccine provides immunity against measles, mumps, and rubella. During an outbreak of a disease, vaccinated people will remain healthy. As they won't become infected, they won't pass the disease on. When most of the community is vaccinated, it makes it difficult for the disease to spread within the community. This is called herd immunity. If more people within the community remain unvaccinated, herd immunity can break down, allowing the disease to spread more easily. Herd immunity is especially important for individuals in the community who can't be vaccinated. For example, if they're too old, too young, or have weak immune systems.
So that was just a quick, simple video kind of showing the need for herd immunity, because if we can get enough people immune to COVID, then we slow the spread of the the virus. So let me just go back to my slides. So for COVID, it's estimated we need to get to about 70 to 80, 85% of the population. They need to become immune to COVID for us to reach that herd immunity level where the virus really can't spread through our communities because there's enough people who are either vaccinated or have had COVID, and it really slows down the spread in communities. Right now, it's estimated about 30% of the population has had the infection. Uh, it's hard to know the exact number. A lot of people are asymptomatic or they never had symptoms, but they did have COVID. Um, so, so right now we're about 30%. So we still have 40 to 50% of the population that still needs to either get a vaccine or become infected with COVID to get immunity. And obviously, we don't want people to get infected. We want them to get vaccinated. And once we get to that level, it will really slow down spread so much that a lot of things hopefully can go back to normal. Something important to point out, right now the vaccines aren't approved for children, so that's a big portion of our population we're not going to be able to vaccinate yet. There's trials underway, so hopefully in summer or um, sometime in the future, those vaccines will be approved for children, but right now it's just primarily adults. Um, but if you think about really getting to that 70 to 80%, so the majority of our population has to be immune. We really need to vaccinate a lot of adults to reach that level. So vaccination is super important, especially with the COVID-19 pandemic, and for a number of reasons. One, the vaccines through their clinical trials have shown to be really effective at preventing new infections. So the Pfizer, the Moderna, their clinical trials had really high rates of showing ability to prevent new infections. Some of the new ones, the Johnson & Johnson and other vaccines, while they have lower effectiveness rates, they still have been pretty effective. Secondly, COVID-19 can have serious life-threatening complications, and you really don't know how COVID will affect you. You might meet a lot of people, they tell you they had COVID and it was no big deal, but in reality, you don't know if it's going to be serious for you unless you get it and see what happens. So without really knowing, the best thing to do is use these preventative measures and then get vaccinated when you can. With these vaccines, they also, in addition to preventing COVID-19, they also can reduce your symptoms and reduce the risk of getting seriously ill if you do get COVID-19. So this point has been really highlighted with the new strains of COVID. We're not exactly sure how well the vaccines work, but we do know that the vaccines most likely will reduce your symptoms. And let's say under normal circumstances, you were going to get hospitalized, but you got vaccinated and a new strain comes, you might get sick, but you might not get hospitalized. So it really reduces those symptoms, frees up our hospitals, and it can really have a good positive impact on the pandemic. Again, pointing out that getting vaccinated doesn't just protect you, it protects other people, the herd immunity, the most vulnerable people in our populations. So you really want to do it for both yourself and other people. And if you do get vaccinated, it can be a way to slow new strains from emerging, slow the number of mutations. We're seeing some new strains of COVID around the world. So I just wanted to go over a few myths before I turn it over to the next speaker about vaccination, and they might touch on these as well. So the first myth, a lot of people think that if you get a vaccination, it can infect you with COVID-19. And the current vaccines we have, none of them have live virus. And they'll go over, the other speakers will go into details how the viruses or the vaccines are created, but there's no live virus, so it can't give you COVID-19. But if you do get vaccinated, you might have side effects, but you will not develop COVID-19 from the vaccine. Another question I get a lot is, if you've already had COVID-19, do you still need to get a vaccination? And the current recommendation is yes, because we don't really know how long the natural immunity from being infected will last. And it seems like the vaccine, it will just strengthen your immune system and make it more able to fight off new infections. So it is recommended to still get the vaccine, even if you've had COVID-19. The next one I get a lot is it safe for me to get a COVID vaccine if I want to have a baby? And all the data says, yes, this should be safe for women to get, and they should still be able to have babies because it won't impact their fertility. And the last one is, do I need to wear a mask after I get vaccinated? So yes, because we aren't sure how likely it will be for you to spread COVID, even if you are vaccinated. That's one reason. The other reason people don't think about, you have to get 2 doses of the current vaccine, and those are 4 weeks apart. And then immunity probably takes at least 2 more weeks. So it's not like you can just get your vaccine and quit wearing a mask. You should keep wearing your mask. One, you won't really have immunity until at least 6 weeks after that first shot you receive. And then we just don't know how likely it is you can still be spreading the virus even though you're not getting infected with it. So those are kind of some of the big myths I just wanted to touch on. So thanks for your time. We'll have questions at the end, and I'm going to turn it over to the next speaker.
Thank you so much, Dr. Hunt. We will move on to the next topic. Again, if you have any other questions, put them in the chat box or the Q&A box, and we will get them to the correct panelists at the end. Our next speaker is going to talk about vaccine technology and development for COVID-19. That's Dr. Alan Young. Dr. Young received his PhD in immunology and pathology from the University of Toronto. His work as an immunologist primarily focuses on domestic animals and the response of domestic animals to zoonotic diseases, and also applications of animal models for new therapies and diagnostic techniques for human disease. Dr. Young's also the founder of MedGene Labs, where he works to assist in the translation of early stage vaccine technologies to the marketplace, and to also develop new tools for human and veterinary medicine. In addition to that role, he also is a professor and research leader here at SDSU. So I will pass the mic to Dr. Alan Young to talk about vaccine technology.
Okay, thank you. Yeah, I think that the first speaker did pretty well covering a lot of stuff, so I will move fairly quickly in, assuming that this works. Okay, hopefully you can see the screen now. Give it a second to kick in. There we go. So my job is basically to talk a little bit about how vaccines are made and how they work inside the body. Oh, good, it's got a timer. So basically, when we're trying to make a vaccine,
Dr.
the question—
Young, I'm so sorry to interrupt. I don't
Yep.
see, can anyone else see the slide? For me, it's just a
No.
white.
Just a white thing. Okay.
So sorry.
That's okay. I've got 3 screens going. That's probably not happy. Give me a second.
No worries.
It's okay.
It's important information. I want to make sure everyone can see it.
Let's try that again then. Okay. All right, can you see it this time?
I can see it within the PowerPoint presentation. So it's not full screen, but we can see it.
Let's see if full screen kicks it off or leaves it. Now see it with the full screen?
Yes, good to go.
Good. Okay, so when we're making a vaccine, and COVID is no different, we're trying to inhibit the ability of the virus to replicate inside you, basically, to break down its ability to infect and to cause disease. And so there are basically 2 targets that the immune system can take. So when we look at how the virus itself infects, everybody's seen pictures of the coronavirus with the COVID. The virus itself is surrounded by this protein called the spike protein, or the S protein, that allows it to stick to the cells of the body through this thing called the ACE2 receptor. That allows it then to enter the cell, where it then starts to replicate and make more of itself. So if we wanted to actually inhibit the ability of this thing to cause disease, there's a couple of different routes we could take. First is we can block the ability of the virus itself to attach to the cells and to get in, or we can get rid of the ability of the virus to replicate inside the cell. And in fact, that is what the immune system does. It has two components. The first are these cells called B cells. And what they do is they make these things called antibodies. Antibodies are proteins specific for different targets. And in this case, what we want to do is make antibodies against that spike protein. Once these antibodies are made, those antibodies bind to the surface of the virus, and they actively block the ability of the virus to bind to that ACE2 receptor and to get into the cell. So we don't need to use the whole virus in order to generate this immune response. All we need is this spike protein presented in the right way to the immune system in order to generate these antibodies. And when we talk about using convalescent sera, we talk about serum titers. You might have heard that. What we're actually talking about is the presence of these antibodies. Now, the immune system also has other cells known as T cells. And what these guys do, you can also call them killer cells. They take care of the other half of the equation. So if we can't get, so that we block the ability of the virus to actually get into the cell, what we can do is identify the cells that have become infected and kill them. And that is literally what T cells actually do. So when we talk about the nature of the vaccines that are on the market, what they're all doing is they're attempting to deliver this spike protein all by itself without the rest of the COVID virus at all, and induce that immune response so that you produce large amounts of antibody, and in some cases produce some of these killer cells as well. We're going to talk about two different kinds of vaccines that are coming, that are either here or very shortly will be coming. The first are the mRNA vaccines from Moderna and Pfizer that you've heard about. They excel at this side of the equation. They're so-called killed vaccine products, or they're non-replicating, which means that they're good at producing antibody responses against the spike protein, and we'll talk about how that is done. And then the Oxford vaccine that you may have heard about from AstraZeneca, and the coming J&J, Johnson & Johnson vaccines, they use a different delivery mechanism that is closer to a live type of challenge. And that produces a few more of these T cells that are active in the immune response as well. And so that explains at least partially the difference between these two formulations. So all vaccines work by trying to simulate a natural infection. What we want to do is we want to build up that immune response against the spike protein or against whatever the target is in that particular vaccine without actually giving you the disease. And so once we've done that, there's two aspects. You'll build up those antibodies to protect you against infection, or those T cells, but you will also generate what are called memory cells. And these memory cells hang around for a various period of time, able to respond more effectively the second time you get exposed to it, and to boost that. And so one of the things that you'll hear about is we'll talk about booster vaccination, and that's really targeting these memory cells in order to give you a long-lasting immunity. So one of the aspects that we deal with in making vaccines, obviously, is we work to try and minimize any side effects of the vaccination process. Anybody who's had to take their kids in for the annual influenza vaccine knows that there's the stick in the arm, and then there's the sore arm a little bit later on. And one of the things to realize is that the more that a vaccine can mirror a natural infection in general, the stronger it is. And so that sore arm you get, or the response that you get, is actually a good thing because it's provoking a good, strong immune response. It is not giving you the disease. It's because, again, there's no COVID there, there's just a spike. But it is telling your immune system that there's something dangerous, or there's something that it needs to get rid of. I don't like using the term dangerous, but that was coined a long time ago. And so what you're basically doing is fooling the immune system into thinking that it's got a response that it needs to generate as a protective response. Ideally, the vaccine will produce sufficient antibody, which is that serology we talked about, against the spike protein to block the ability of the virus to bind to its receptor. If it's completely successful, that's what we call neutralizing immunity, which means that the virus never actually infects you. You get it in and it gets cleared. Very, very few vaccines produce neutralizing immunity. Far more common is you have enough antibodies that the antibodies kill off the virus before it has the chance to cause significant disease. As well, cellular immunity, that T cells that I talked about that actively kill the cells that get infected with virus, is actually a good thing because that takes care of any cell virus that does get into cells. So the mRNA vaccines produced by Moderna and Pfizer are basically the same thing. And what they take advantage of, let's see, I'm gonna get rid of, I think this might be blocking something. What they take advantage of is the fact of how our cells and how viruses replicate in cells. So going back to elementary school biology, we learned that DNA is how we code for all of our proteins. We pass that on in our cells. That becomes messenger RNA or mRNA, and that eventually gets turned into the proteins that make our cells work. And spike is a protein like anything else. And so what these vaccines do is they figure out a way of getting the messenger RNA for that spike protein and only that spike protein into the cells. The cells of your body then produce that spike protein, which then is recognized by the immune system and generates the production of the antibodies that we talked about. In order to do this, they produce the mRNA in a lab reaction. It's completely cell-free. They make this RNA into a sequence, and then they package it inside a lipid membrane, just like you would your cells or anything else. And this lipid membrane allows it to fuse with the target cell, get inside of it, and make lots and lots of spike protein to stimulate the immune system. So just like all of the other vaccines, the question is, how do you make the immune system recognize that spike protein? In this case, the mRNA vaccines, when they inject, make your cells produce that. The mRNA itself doesn't stick around for a terribly long period of time. It's not capable of replication, so it goes away eventually, but your antibodies will remain. It's important, as people talk about the safety of this, to realize that animal health companies have been selling RNA-based vaccines since 2014, when we had a coronavirus outbreak in pigs. And Merck is currently marketing these in hundreds of thousands, actually millions of pigs, with no adverse effects. This is a routine vaccine that's on the market. Johnson & Johnson and AstraZeneca, this is a slightly different way of making the target protein. In this case, what they've taken is an adenovirus, which is a common cold virus. They have taken away one of the genes of the adenovirus and replaced it with the spike protein. So you, again, don't have an active coronavirus. You have the common cold adenovirus that is now expressing that spike protein, and that's how they get that into the cell and into the immune system, is by giving this virus. The way it's produced is that it is grown in cell culture in cells in a lab, basically, or in a big fermenter. It requires what's called a helper virus, and at the end of the day, what you get is a released virus, which is an adenovirus, but now is expressing these spike proteins on the cell surface. So it looks on the outside a little bit like the COVID-19, but that's all. The inside is not the same. It has also been modified so it can't replicate after it gets into your cells. So even though it's a live virus vaccine, it gets into your cells, it makes the spike protein, but then it dies off. It can't move any further. And so one advantage of this is this gives, even though it's only at a lower level of protection relative to the mRNA vaccines, it's a one-dose vaccine product. So it's able to stimulate strong immunity with a single dose as opposed to a double dose, because it is a live vaccine product. These two have been in use in animals and have been developed for humans as well. A common wildlife rabies vaccine is based on an adenovirus platform. One of our FMD vaccines for animals is also in the same factor. So these are not really an unknown commodity either as far as safety, and they have been used in the field widely. So I'd be remiss if I didn't talk a little bit about side effects. Everybody's heard about the vaccine side effect issue since 1998, when a discredited scientist named Andrew Wakefield tried to drum up some personal income by promoting theories about the MMR vaccine, which has been difficult to shake off, but that wasn't the first. The very first vaccine was actually the one that was developed by Jenner that eliminated smallpox. And in order to vaccinate that against the smallpox virus, he found that he could use a cow virus called cowpox, which does not cause any of the problems in humans that the smallpox virus does. When he gave it to people, they were then protected against smallpox, which was a devastating disease. But of course, even back then, you probably can't see unless you've got a big screen, but people were convinced they were going to turn into cows or have cows growing out of their arms or out of their noses or whatever as a result of taking that vaccine. And clearly that doesn't happen. And so while you may hear about a lot of this, the vice president of operations at our vaccine company has got 40 years in the vaccine business. And this is one of his favorite phrases: the plural of anecdote is not data. And what that means is just because a lot of people say something doesn't necessarily substitute for careful scientific study and evaluation. And what I use in my class is the reference of jumping out of a plane without a parachute. In 1972, a Serbian flight attendant was in an airplane that had a terrorist bomb go off and explode the plane to 30,000 feet. She fell with the plane and she survived. And it holds the Guinness record actually for the longest free fall without a parachute. Just because this happened, and a lot of people talk about it, and clearly you can survive at 30,000 feet without a parachute, I still think I'd want a parachute if I were to jump out of a plane, and vaccines are no different. So at that's all I actually have. I will unshare and I'll let Natalie take over. There it is.
Thank you, Dr. Young. Our last but not least panelist, many of you might recognize. She has already done a few discussions about the vaccine and safety efficacy for the city that can be found on YouTube. So if you've seen those, you would probably recognize Dr. Natalie Takes. She is also an assistant professor here at SDSU. She completed both her Master's of Public Health in Hospital and Molecular Epidemiology and her PhD in Toxicology at the University of Michigan. Her specialty areas include mammalian cell biology, membrane trafficking, and CRISPR genome screening. Currently, her research here at the Department of Biology and Microbiology is focused on how molecular machines of cell function in normal versus disease states. So she will apply that research to the next topic, which is vaccine safety and efficacy. Dr. Takes your.
Yep, I got it. Thank you. Hi, everybody, and thanks for coming today. Like Laura said, I have an interesting background that covers both epidemiology and cell biology. So my current research mostly focuses on immune cell biology. And I've also done over the years a number of serology studies looking at the seroprevalence of antibodies to viruses. So most recently, my lab has worked on testing healthcare workers here in Eastern South Dakota and Western Minnesota to see what percent of them have antibodies to SARS-CoV-2, the virus that causes COVID-19. So that's my current project and how it relates to our talk today. But mostly for the slides I prepared for today, just focus on the clinical trial data that allows us to determine the safety and the effectiveness of the currently authorized COVID-19 vaccines. So I was kind of talking to some colleagues this morning, trying to sort of see what is the major hesitation that people have when considering these vaccines. And I kind of came to the broad conclusion that I think a lot of people's uncertainty and hesitation might stem from the pace of the vaccine development and authorization process and the distribution. So normally vaccine development, testing and distribution takes a very long time, a decade or even more. So this is a timeline from the New York Times showing that. Normally we have to test, develop the vaccine, test it in cell culture, test it in animals, start with small human trials, then get to bigger human trials, and then even bigger human trials. And then only after it's shown to be safe and effective in the phase 3 randomized control trials, then do we get to the point of building the factories and manufacturing the doses to distribute to the population. And because COVID-19 pandemic is such a huge public health issue, this process has been drastically shortened. And I want to point out, it's really important to realize that all of the same phases that were there in the sort of traditional timeline are still here and still have taken place. It's just that they've been done simultaneously instead of sequentially. So while the academic research was going on, the early clinical trials were started. Then instead of waiting for the end of the first early clinical trials, the second and third phases were started very shortly afterwards, so that all of these things could happen at the same time. Now, this is a huge gamble, right? And a cost to both the government and the companies doing these things, because if they get to the end of phase 3 and find that it wasn't safe or efficacious, then having built a factory for this process would have been a waste of money. But the companies that are doing these trials and the federal government was willing to take that chance because the public health problem is so dire and needs to be solved. Because as you saw the data that Dr. Hunt showed, almost 500,000 people in the United States have already died of this virus, right? So it's really a public health problem like we haven't seen in a very long time. So I'm gonna jump right into the phase 3 randomized control trial. So these are the these trials provide the data for how we determine whether the vaccines are safe and effective or efficacious. So in order to be able to trust the data, you really need to understand how the trials were conducted. So basically, and I'm going to use the Pfizer vaccine as the example here. Both the Pfizer vaccine and the Moderna vaccine are currently authorized by the FDA through an emergency use authorization. And like Dr. Young said, they're very similar vaccines. They're both RNA vaccines. But I'm just going to talk about Pfizer and mention Moderna briefly, since they're very similar and it might be confusing to try to keep them both separate. But at the beginning of the trial, participants, human subjects, are recruited who are willing to volunteer, and they are randomized to either in the vaccine group or the placebo group. So they're separated based on their age and their comorbidities or other diseases they have, their race and ethnicity and so forth. So each of the vaccine group and the placebo group would have very similar demographic characteristics. And after they're randomized, they're given their first shot. For the Pfizer vaccine, we wait 21 days and get the second shot. Then we wait 7 more days and have the participants monitor themselves, both for side effects of the vaccine, they're listed here under safety, as well as symptoms of COVID-19. So if a person in the study gets any of the COVID symptoms like fever, increased cough, shortness of breath, muscle pain, loss of taste or smell, or that kind of thing, then they report that to the study personnel and get tested for COVID-19. Then if they are found to be positive based on an RT-PCR test, the gold standard for diagnosing COVID, then they're categorized as having got the disease. And we can go and compare the percent or number of people that got COVID who've been vaccinated and those who got the placebo. The placebo is a saline injection, so it's just salt water. But the person gets the shot, so they don't know if they've gotten the real vaccine or the placebo. So when they're monitoring for symptoms, they don't know if they've received the real vaccine or not. At the same time that they're monitoring for symptoms, and even during the 7-day period following the second shot and the first shot, they're monitoring for other side effects, local side effects, things like injection site pain, redness, swelling, swollen lymph nodes, fever, and so forth. And they also report those to the study. So basically what we can do after this is we can compare the number of people who got COVID in the vaccinated or placebo group and the number of side effects in the vaccine or placebo group. And from this data, we can determine whether or not the vaccine works to prevent infection or prevent disease actually, and whether it has any burden of side effects. One of the things that I want to point out from this study design is that these symptoms that are being monitored here are for the sign of COVID disease. So this data will tell us if the vaccine helps prevent COVID disease, but it does not tell us if it prevents asymptomatic COVID infections. So therefore, that's the reason why you still have to wear a mask even after you're vaccinated, because there's nothing in this data that tells us if a person who's vaccinated stops shedding virus, okay? And I want to point out that the time when this data was reviewed by the FDA, the follow-up was about 2 months. Now that was back in December, and the study is continuing to go on so that we'll continue to keep following people in the study for 2 years total, unless they choose to be unblinded and vaccinated. So this is the way that the data is presented to the FDA, and it's kind of complicated, so you don't have to look at all of it, but I want to point out to you that this group here shows the number of people who got the vaccine. So about in the Pfizer trial, about 18,000 people got the vaccine, and the same number of people got the placebo or the saline shot. And then they were followed following the administration of the second dose. And of those who got the vaccine, 8 got COVID. But of those that got the saline shot or the placebo, 162 got COVID. And if you break it down by age group, you can compare the number of people who got COVID in younger folks, ages 16 to 55, and those who are 55 or older. So the important thing to notice right here is the calculation of the vaccine efficacy, and it's 95%. This yellow text here at the bottom shows how that's calculated. But a simple way to interpret it is that in the people, so 95% of the people who got the COVID vaccine who would have normally gotten COVID were protected from it. But note that about 5% of people who normally would have gotten COVID based on the data from the placebo group were not. And this is one of the reasons why Dr. Hunt was trying to emphasize the concept of herd immunity. Vaccines are never 100% effective. And the reason why all of us have to get vaccinated, or most of us have to get vaccinated for herd immunity, is to protect those small percent of people for whom the vaccine doesn't work, as well as for the people who can't get the vaccine because of for various reasons. This is another way of looking at the data. It's a little bit more friendlier to look at if you're used to looking at graphs. So on the x-axis is the days after the first dose. So you can see that this data starts at day 0. And on the y-axis is the cumulative incidence of COVID-19 occurrence. So basically at the beginning of the trial, after the placebo recipients as well as the vaccine recipients got their shot, they were monitored to see if they developed symptoms of COVID and then tested if they had any symptoms. So in the very beginning, you can see that the vaccine didn't offer any protection, and the people in the vaccinated group for the first couple of weeks of the trial had basically the same rate or incidence of COVID-19 as the placebo recipients did. But after a couple of weeks, it leveled off so that very few people in the vaccinated group continued to get COVID, whereas those in the placebo group had received, who were exposed to COVID, developed symptoms of the disease. So based on this, I just want to emphasize how completely excited those of us who were following this data were to see how amazingly effective these RNA vaccines were at preventing COVID disease. And if you were to ask me, back in, I don't know, early fall, what I was worried about, I would have told you that I was worried that the vaccines weren't going to work that well. So we were all so excited to see how amazingly well these vaccines worked. And I think that's kind of a take-home point here, that we're just really lucky that they're working as well as they did. So, but a lot of people have been asking a lot of questions about how do we know they're safe? And I want to point out how we know they're safe is because we compared the side effects in the people in the placebo group to the people who were in the vaccinated group. And side effects are divided into 3 basic categories: local events, systemic events. So local events are things like pain, redness, and swelling at the injection site, in the arm, right where you got the shot. Does it hurt afterward? Systemic events are things like fever and tiredness, which are things that you feel throughout your whole body. The last category of side effects are called I'm going to forget it. It's in the next slide, but adverse effects, things that are beyond the normal vaccine side effects. So both the local events and the systemic events are side effects that we expect based on our knowledge of the immune response. And Dr. Young kind of alluded to this. If you get a vaccine, you actually want your arm to hurt afterward. That's the pain that you feel in your arm is a sign of your immune system recognizing the antigen and mounting an immune response against it. So it's actually not a good thing if you don't have pain, redness, or after you get a vaccine. Just a little side note on the way that our immune system works. If you get an infection and you feel sick, some of that's because of the way that the infection is affecting your cells, right? But a lot of the sickness that we feel from infection or from a vaccine is because our immune system wants us to slow down while it fights off the virus. So our immune system actually makes us feel sick so that we, take a nap instead of trying to work, for example, when we're sick. So pain, redness, and swelling, first of all, you can see that they're much higher in the vaccine group compared to the placebo group. Pain is the most common side effect from a vaccine, pain at the injection site. And what this is comparing is the younger age group, those aged 16 to 55 after the first dose and the second dose, compared to people over age 55 after the first dose and after the second dose. So what you can see is the younger folks have a little bit more side effects than the older folks, and that after the second shot, well, in the Pfizer vaccine, there wasn't much difference between the first and second shot, but in the Moderna vaccine, there's a little bit more side effects after the second shot. One of the reasons why younger folks probably have more side effects than older folks is because they have a more robust immune system that reacts better to the vaccine. So that's one of the reasons why when a person gets to a certain age, for example, they have to have the old person influenza vaccine because their immune system doesn't have as robust of a response as a younger person's would. This is a graph that shows the systemic events in the younger age group after the first and second dose of the Pfizer vaccine. The most common side effects that we see in both the young group and the old group are fatigue and headache. But I want to point out here that a lot of people in the placebo group also have fatigue and headache. So I take that as a sign that a lot of us are walking around feeling kind of crummy frequently. Other things besides fatigue and headache are some people have fevers and chills and muscle pain and joint pain. So, and then you can see that the use of antipyretic medication, that's medication that lowers fevers, is a little bit higher in the vaccine group compared to the placebo group. I just want to emphasize that all of these side effects are expected based on how we understand that the immune system responds to the vaccine. It's a sign of the immune system responding to the vaccine. Those side effects are a little bit lower. As I mentioned before, these systemic events are a little bit lower in the folks who are older than age 55. And then again, that's probably because they don't have as robust of immune response as the younger folks do. The last, so I just want to conclude with a repeat of what I've just said. The local reactions to the Pfizer mRNA vaccine were mostly mild to moderate in severity and resolved within 1 to 2 days. The systemic events were reported more often by younger vaccine recipients than older vaccine recipients. And the most commonly reported systemic events were fatigue and headache. The adverse events are very rare, and I didn't even make a graph of these because they're so rare, it's kind of hard to look at them. But 64 vaccine recipients, or less than 1%, reported swollen lymph nodes, or some participants there were very few participants altogether in either group that had severe adverse events. But 4 related serious adverse events were reported among the vaccine recipients. So that was a shoulder injury, lymph node swelling, ventricular arrhythmia or heart arrhythmia, and paresthesia in the leg, that's like tingling in the limbs. Of the people in the trial, 2 people who received the vaccine died, one from atherosclerosis or heart disease and one from cardiac arrest or a heart attack. And 4 placebo recipients died, 2 from unknown causes, 1 from hemorrhagic stroke and 1 from a heart attack. So what you can see is that you have to be able to compare the people who got the vaccine to the placebo in order to be able to tell if the things that happen are due to the vaccine or if they're just things that would normally happen anyway. And so when everyone's asking, how do we know it's safe? My answer is, this is how we know it's safe, because we gave the vaccine to equal number to people in the phase 3 control trials, and we gave a placebo or a saline shot to the same number, and we compared them to see all of the different side effects they had. And we also compared them to see how many of them got COVID. So I will just answer a few questions that I saw. One of them was about if children can be vaccinated, and so I will point out that right now the Pfizer vaccine is approved for people who are over age 16. And the reason why it's not approved for children is because they weren't included in this phase 3 trial, right? It was only people aged 16 and older. And the other question was whether or not, and one of the things to keep in mind is that vaccination is always a cost-benefit analysis. Because kids have a less severe COVID disease, there's not as much push to get them vaccinated, given that they haven't been included in trials. The other question was, should pregnant women get the vaccine? And right now, the USA definitely encourages pregnant women to get the vaccine. And there's not any data to suggest that it's not safe for pregnant women. Pregnant women were not included in the trials, partly because the animal trials that are necessary to include pregnant women in the trials were not finished by the time these phase 3 trials started. But the animal studies have subsequently been finished, and there have been no signs in those experiments that there's any risk for pregnant women different from the rest of the population to get these vaccines. So that basically concludes my part, and I'm happy to take any questions.
Thank you so much to all of our panelists. We have a lot of questions, and I don't know if we'll get through them all, but I will start at the top and we'll go through them. The first question that I have in the Q&A box, I don't know if this would be for Dr. Takes or Dr. Young, but it's how many COVID vaccines are there and what's the difference? I know you've both talked about a couple, but is there just a really brief overview you can give?
Well, I want to say right off the bat that there are 2 that have emergency use authorization from the FDA. And Dr. Young mentioned a couple of other vaccines that are in the pipeline. They are currently in phase 3 trials, but they have not been, their data has not been reviewed by an outside source at this point in time. And the last I saw, they weren't expected to be reviewed by the US FDA until later in the spring.
Yeah, there's dozens in the pipeline, but there were 9, I think, or 10 in the original Operation Warp Speed program. Some of those have been abandoned for various reasons, but probably one of the more common reasons is they weren't first out of the gate, so they commercially were not viable. I see that as of today, the Novavax vaccine, which is another one that's coming, is in phase 3, and the University of Minnesota is participating in that one. So over the next 8 months or so, you'll probably see other ones coming out, including like, I don't know whether they're going to pursue the Oxford vaccine at the FDA level or not. It's in Europe right now in use. The J&J I would expect to see come through, and I wouldn't be surprised to see Novavax push all the way through. So there are other ones, but they're not available now, and as a result of not having the emergency use authorization, they are quite a ways out yet.
Along that same question, there's also one about whether or not one vaccine is better than the other since they're produced differently.
No, they're almost interchangeable as
That's
far
why
as
I
the two
send it
mRNA.
Pfizer data, because their data is so similar between them.
Awesome. This question specifically is for Dr. Young. It says, how long after recovering from COVID should a person wait before getting the vaccine? Will circulating antibodies interfere with the vaccine ability to stimulate immunity?
Well, I think it's actually more than just for me, because that's part of a public health question as well from an analytics. I can say that based on what we have seen, you probably would not want to take the vaccine while it was still circulating virus, but that there'll be health data as far as that goes. The chances of getting a block or an inhibition as a result of the circulating antibody titer from what's happening with COVID. One of the challenges with COVID is it tends not to boost to really high antibody titers through natural infection, which is why we believe that reinfection is a possibility. So I honestly, I'm going to defer to the public health authorities as far as what they've actually seen as to how long you should wait before getting the vaccine afterwards. But from an immunological perspective, it's not a huge concern.
There was a study out last week that showed that getting the vaccine after getting the natural infection caused a nice bump in the antibody response. So that's a good reason to get it, even if you've already had COVID. And given that the vaccine distribution is so logistically challenging right now, it seems like the best time to get the vaccine is when you have the opportunity to do it.
Exactly.
And it's not, don't think much harder than that. Yeah. If someone offers you the vaccine, take it. Because I will just mention, I have my data from my seroprevalence study. We do see pretty low titers from a natural infection. So I don't think that we should really count on natural infection in providing any semblance of herd immunity. We really need the vaccine to do that. I noticed that Dr. Scholl had asked a question on, is it possible to get a better antibody response from a vaccine than from natural infection? And yes, there is data out there now showing that the vaccines give the highest level of antibody response. The vaccine titers are in the upper quartile compared to the natural infection folks.
Yeah, there's fairly strong data out that the virus itself is inhibiting the immune response during infection. You don't get that with a vaccine. You're just getting the protein, so that's gone. So I saw one question on there. Maybe I should wait, but about animal vaccines. Why can't we use animal vaccines in humans? The short answer is that the vaccines that we use in animals and the ones we use in humans are virtually identical. There's a couple of differences in terms of the components that go into it, but by and large, they're basically the same thing, as I said. However, what Dr. Teeks went through is the regulatory procedure to go through. So just because we know that you've done millions of pigs with this vaccine, there is still the possibility that there's going to be some adverse events in humans as a result of allergic response to some of the components or things like that. So you still have to go through that regulatory process, that testing process, in order to adapt those. There have been suggestions of going the other way, that are going on right now, and that is using some of the human vaccines in the animals, particularly the susceptible species. The same rule applies. You can't just make that jump because there are different concerns in animal vaccines than there are in human vaccines, notably at the ecological level or the environmental level that you just don't have to worry about with the human side. So there really are two parallel but independent pathways. That being said, the knowledge of how to make the vaccines is there. And I guess the other thing I would say is, just because we have coronavirus vaccines for animals doesn't mean that they're specific for the COVID protein. We have lots of coronavirus vaccines for lots of different coronavirus families, but unless it's specific for the COVID spike protein sequence, it's not going to work. So even the original SARS, one of the reasons that we have Moderna and Pfizer and all of these mRNA vaccines is because they have spent an exceedingly large amount of time at a research level as developmental companies working on SARS as a target. So they already had all of this in their pipeline. All they had to do was swap out the SARS sequence and put in the SARS coronavirus 2 COVID sequence to move forward. But they couldn't just use that SARS vaccine that they'd already had in the pipeline. They had to make the new one to go ahead.
Yeah, and that kind of segues into one of the other questions that we were asked to answer even before we started, which was, what are the long-term effects of this? So I would point out that there are a number, like 17 other clinical trials for non-COVID RNA vaccines in humans. So we already have some data on side effects just from the technology, right? The lipid technology that's used to package the RNA. When people ask about long-term effects, I have sometimes a hard time answering that question because I don't know what they're asking. I think people are wondering, is there anything that could happen to me a year or two from now that would become because of this vaccine? And first of all, I want to point out that we know a lot about now the long-term effects of COVID infection and disease itself. That is not up for debate. And there are some very serious long-term effects from getting the disease. As far as the vaccine goes, we get our data from the clinical trials. In addition to that, everybody who has gotten the vaccine has the opportunity to report their side effects to the federal government through a vaccine adverse event notification system. So we continue to collect data. Sometimes people call it phase 4, right? Even when it goes to the large scale population administration. But keep in mind that most side effects from vaccines happen within hours to days of getting the shot, right? That's because the immune system is responding right away to the injection and to the antigen that was put into your body. There have been historically some long-term effects, such as something called Guillain-Barré syndrome. It's an autoimmune disease that can result, it mostly results from infections like influenza. And it happens 2 or 3 weeks following the infection. Historically, there were a few vaccines that had a little bit higher rate of Guillain-Barré in the vaccinated population compared to the unvaccinated. But I want to point out that the rates of Guillain-Barré in vaccinated folks is still less than it is in people who get the actual infection, and that those adverse events happen a few weeks following administration of it. So as far as long-term side effects go, we don't see any data in the current clinical trials to indicate long-term side effects. Furthermore, in the 70 million or plus people that have currently been vaccinated, there hasn't been any indication of a problem. So I feel very comfortable saying that there is very little evidence of a long-term side effects from these vaccines. And compared to COVID itself, it's not even on the same page for comparison.
Thank you. I will move on. So we have a couple people asking about additional resources for the Operation Warp Speed program. Does anyone have a recommendation of where individuals should go if they want more information on vaccines?
So I'm not sure if Operation Warp Speed itself put out a number of news releases, and you can do that with a Google search. I'm not sure if those are still active or not, given certain changes. Do you
The
know, Natalie?
CDC website is very informative, and it should definitely be your first source of information about the vaccines. So before you read any other things on social media, please consult the CDC. It's very nicely communicated with easy to understand infographics and information. And the goal of the CDC is to protect the health of our population. That is their number one goal, and they're experts at doing that.
We have another question about the Pfizer study specifically, asking how the group was exposed to the virus in the exposure arm.
The group were just exposed naturally. So if in their normal day-to-day life they were exposed, that was how they were exposed. So there was no human infection. It's not necessarily ethical to do that.
All right.
But I will point out that there were a lot of animal studies that preceded that, where they did give the vaccine to animals and did expose them to the virus. And the other thing that I didn't mention, but that in the earlier phases of the human trials, there were also a lot of blood tests to look at neutralizing antibodies in people who got the vaccine. So before we ever knew that it prevented the disease, we were guessing that they would do pretty well because of the nice neutralizing antibody response that was seen in the participants.
Right, so I'm seeing a couple questions about the timeline for SDSU individuals and what that would look like for employees at SDSU. So I just wanted to add that SDSU employees right now are in 1D with educators, and that the most up-to-date information about that can be found on the Department of Health website. And right now, South Dakota Department of Health timeline estimates that would begin no earlier than March of this year. So we'll continue to update, but that's the timeline right now based on the Department of Health information. We also have another question that is a little bit longer, so I'll try to read it. So this says that if you're vaccinated, it's a question, if you're vaccinated for a disease and immunity wears off and you eventually do contract that disease, will you get a more severe case of that disease than if you had never been vaccinated?
The short answer is the reverse is likely. Even though your antibody titers and things may wane over time, the fact that you already have been exposed gives you those memory cells that we were talking about that are prepared to take effect more effectively the second time around. So that being said, we haven't talked about, I think I would feel safe saying that while our goal right now is to get everybody vaccinated and to knock this thing down as much as possible, we're probably looking at this becoming a yearly vaccine for a while, similar to the influenza, in order to stay ahead of it.
All right. Well, since we won't be able to get to all of the questions, I did want to touch this one. I think it'll be interesting. It's, once COVID is finally settled, how long do you believe it'll last in our world?
Viruses don't go away easily. I would defer to everybody else on that one, but it will eventually become something I think that we will have under control, closer to the ones that people are more familiar with, influenza and some of those other ones out there. That's my feeling, but I will defer to the other panelists for theirs.
Yeah, I'll agree. You know, the one, or you can think of smallpox. That's one, virus we eradicated, but it was super difficult. You needed global response, and it's a totally different virus. So I would agree. It'd be more like influenza unless there's a lot of advances in the future.
So just keep in mind that one of the most challenging aspects currently of this virus is the new mutants that have arisen. And in order for a virus to mutate, it needs to replicate. So the more infections that we can prevent, the more that we can prevent new mutants from arising. So as a global community, as a local community, we have to do everything we can, from wearing masks to getting vaccinated to continuing to distance and be careful in social gatherings, to prevent the, new mutants from arising. I will say that this virus doesn't mutate or have the potential to mutate nearly as much as influenza does. So hopefully it's a lot easier to contain than influenza. But right now, when we have billions of susceptible people in the world, that's a lot of opportunity for the virus to mutate, right? And I will just mention, Aaron mentioned that he estimated 30% of people had been infected. In my seroprevalence study in healthcare workers and people, including here in Brookings County, we found a 15% seropositivity. That was from blood that was drawn in November, right? So there's been a few months since then, but that means that 85% of our population is still susceptible, right? And only a few percent have been vaccinated. So there's still a lot of tinder, right? Or to burn up here in our local community and around the world. So yes, vaccination is important, but all of the other things we've been doing are really, really important.
On that same note, you mentioned the new variants that we're hearing. Is the vaccines that we discussed today, are they effective against these new variants?
So there have been a couple of studies on that. The best one's probably on the Moderna vaccine. And the short answer is that there appears to be, in particular the South African variant, appears to be significantly more resistant to, it's different enough from the original parental strain that it's going to be a challenge. And Moderna has actually already announced that they're working on a vaccine to target that. Hence my discussion, this is likely to turn into something like influenza where we'll get a boost. Now, it's entirely possible that there'll be enough cross-reactivity that it will knock back a little bit the infection, so it won't be as severe. We have yet to see that, but we do know that those antibody titers we're talking about are not as cross-reactive as we would like against the South African variant. The good news is, as near as we can tell, the UK variant and some of the other ones that are out there are taken care of by the vaccine fairly effectively.
So keep in mind that the number one, as far as I've read so far, that the most important thing to whether the vaccine or a natural infection provides immunity to the new variants is the person's antibody titer. The higher the titer, meaning the more antibodies you have in your blood, the better you can knock it back, like Dr. Young is saying. Mm-hmm. So people who have had a natural infection could have the same, potentially the same problem as people who are vaccinated if the vaccines don't provide as good immunity to the new variants. But that's just another reason why we need to continue to have sort of the multi-pronged attack on this pandemic, why we need to continue with all of the other measures that we've been doing in addition to the vaccine.
The other aspect, just to finish off, I guess, on this is even though there's not a pathway with the FDA the way there is for animal vaccines for marked acceleration, one of the aspects about the way the Moderna vaccine is made and the way the Pfizer vaccine is made is they're very, very simple for them to change the sequence and update it over time because of the manufacturing method that is actually used. So their ability to get a new vaccine in the field is going to be fairly effective.
Great, thank you so much to all of our panelists. We're already running a little bit over time, but I think we got to almost all of the questions. Are there any closing comments from any of you that you'd like to add to this before we close off the panel?
I was just glancing through the questions. I think, did you answer the questions about availability?
I certainly can expand on it. So right now, for everyone in this state, the State Department of Health gets an allocation, and it's increasing a little bit every week, and that's given to the state, and they're distributing it to each county, and it's run by one singular health system in that county. For us in Brookings County, it is run through Brookings Health System. So we did get a question about whether we would have something on campus. And I would like to echo the comments made by previous panelists that if you have the opportunity to get vaccinated, you should take it. So if you fit into a different category in 1D and it based on age or some other factor, take that opportunity and get vaccinated whenever that is available. So at this time, we're still planning on 1D in that timeline, but because vaccine is still limited until we get widely available vaccine, you should take it as soon as you can get it. Okay, with that, we will close out the panel. Thank you for everyone for coming and attending and sticking out with us for an extra 15 minutes past the hour. If you have any additional questions, feel free to send them our way and we'll get them answered. And if there's one we missed, we'll try to answer on the vaccine Q&A webpage. So thank you, everyone. And this recording will be posted at some point as well. So thank you, everyone.
Thanks, everybody.